afternoon. I'm Stephen Goodman, Chief Scientific Officer for Microscopy Innovations. Chemical preparation is the backbone method for biomedical electron microscopy. This presentation was prepared for the vendor symposium session and for this pre-meeting workshop, now postponed. The goal of this workshop was to discuss innovations to improve chemical prep workflows. I was asked to provide my perspective as a vendor, which I will do in this brief presentation today. I'll review chemical preparation for three important EM applications: transmission EM, immunolabeling, and three-dimensional EM, principally serial block phase SEM. In this presentation, I will begin at the beginning of biological TEM, look at the present, and provide my guesses of the future. The origin of chemical processing EM is the beginning of biological EM, and this began due to these and many other EM pioneers who created the chemical preparation workflows that we build on today, that include aldehyde fixation, osmium post-fixation, solvent dehydration, embedding, sectioning, and heavy metal staining. Here's a look at the EM prep workflow. In the beginning, reagent preparation was done with glass pipettes and glass vials. This has changed little. Today's devices would be recognizable by the EM pioneers from decades ago. I believe this would also include the EM autoprocessor and microwave ovens. The most fundamental process, reagent delivery, has changed little in over 50 years. Let's take a deeper look into the workflow for different specimen prep applications. The beginning of the chemical prep workflow is the initial aldehyde fixation, which is often not in the EM lab. So this is a different workflow issue. This presentation will concentrate on the reagent exchange part of the specimen prep. Here there is a great opportunity to improve workflows. Again, I will concentrate on the reagent exchange workflow for three applications: transmission EM, immunolabeling, and 3D EM. I will quantify the preparation timing and hands-on effort that begins with a specimen initially fixed in aldehyde to the beginning of resin curing. And we'll be able to quantify this over time and effort. So let's begin with manual vial and pipette processing of tissues for TEM. The length of this open bar indicates a typical duration of reagent processing of 2 days. Of course, this varies depending on the specimen in the lab, but rarely is this less than 1 or more than 3 days. The cumulative hands-on labor time is shown by the length of the shaded bar. Of course, this effort is usually intermittent, with no effort during long reagent treatments and a lot of effort during short treatments. Autoprocessors improve the workflow. The total time and labor times are both reduced, with much of the labor being the transfer of specimens in and out of baskets and their documentation. Microwave processing was introduced in the 1970s and refined over many decades, and it provides major increases in prep speed. The workflow is very busy, consisting of manual reagent exchanges in between each microwave cycle. Now I'll look at immunolabeling. Executing these protocols is challenging due to the large number of steps and keeping track of antibody titrations and controls. A typical pre-embedding protocol requires 47 successive reagent immersions to prepare specimens prior to sectioning, while an example of an on-grid protocol requires 28 reagents and each grid to be dipped in and out of 28 droplets. It's easy to make mistakes, damage specimens or grids, and have inconsistent timing, which can decrease the power of the results. For simplicity, this bar only shows the time and then the effort for pre-embedding immunogold labeling, which takes days. In contrast, manual on-grid IGL also requires about a full working day to execute. The last imaging modality I'll examine is 3D EM. 3D EM preparation is like TEM but has even more heavy metal and reactive reagents for longer times. Preparation takes nearly a full working week and requires about a day and a half of hands-on effort. Now I'll move from the past into the present and introduce the MPrep system. I'll first illustrate the workflow for a specimen. A specimen is inserted into an MPrep S specimen capsule where it is then entrapped. The capsule is then attached to a pipetting device, either a manual pipetter or the automated specimen processor, the ASP 1000. All reagent exchanges are then executed through the connection to mechanism. The final reagent is filling with the 100% resin, and then once the capsule is filled, it is then transferred to the oven for resin curing in the usual manner. Manual reagent exchanges may be done with 8 or 12 specimen or grid capsules at a time using multi-channel pipetters, here an 8-channel unit is shown. But for the greatest efficiency and speed, all reagent exchanges, rinses, and agitations are done automatically with the ASP, the automated specimen process. Now I'll examine the metrics for MPrep system workflows. Manual MPrep preparation eases the workflow and reduces effort by enabling one thumb touch, just simply pressing the thumb at the top of the bar, to exchange the reagents on 8 or 12 capsules. But this does not appreciably reduce the total prep time. The MPrep ASP autoprocessor greatly reduces the tissue prep time for TEM with rapid reagent mixing protocols. This provides preparation speeds that are comparable to microwave processing, but the hands-on labor is reduced by about half because the reagent exchanges are done by the ASP robot. The labor mostly consists of setting things up and cleaning up at the end. The reagent prep for both this planarial sample and this muscle sample were automatically completed in about 2 hours. Now let's look at automated immunogold labeling. The data shown here is from the University of Maryland Baltimore Core Lab for both pre-embedding and post-embedding or on-grid IGL. The total time to results was shortened by a day compared to manual prep, and the effort was reduced to about an hour, principally for setup and cleanup. Let's now look at the automation of the 3D EM preparation workflow. Using rapid prep ASP automation, the 3D EM protocol workflow now takes about 1 day instead of nearly a week, and the operator effort is reduced to an hour or so, as reported by the Cleveland Clinic. This Cleveland Clinic work investigated the 3D ultrastructure of brain synapses within 60 micron imaged cubic volumes. This work, done with pediatric clinicians at the University of Wisconsin, acquired 50 micron 3D volumes of heart, and they were used to analyze how mitochondrial shapes are altered in premature infants that spend time in high oxygen incubators. These are the mitochondrial shapes. I believe that this type of 3D image data would have been amazing to the pioneers. I'll conclude with my predictions for the future of the chemical processing workflows. I believe that the future will include the methods of the past due to the wide variety of specimens, labs, and applications. But I think that the advantages of automation will lead to increasing adoption in research and clinical labs due to the high value of faster results with less labor and the capability of following any reagent protocol. And regarding IGL, I think that the daunting labor for manual prep has slowed down the use of this powerful imaging technique since its development many decades ago. Manual and preparation can help, but there's nothing like walk-away convenience of automation. Lastly, I'll look at 3D EM. I think that the difficulty and time-consuming nature of manual prep is slowing down the growth of this powerful new imaging mode. Automation can solve this. Automation could even enable IGL with 3D EM and make something that complicated practical. The future for many applications is automation. So where can automation go? It can be expanded into more protocols and application, which can be shared among users. Controlling reagent temperature has been requested by some customers, which is in place at some customer sites. What do you need? We'd be happy to hear from you. And besides automation, what else? Where do I see the potential? So I believe that there'll be new reagent chemistries, more use and types of molecular labels, improved correlative methods, and I see the initial fixation as a very real problem because it's often done outside the EM lab. And this is something that I hope to be telling you about in the near future. So I thank you for your attention, and I'm happy to take questions through the virtual meeting interface, or you can reach out to me directly or via our website. Thank you so much.